So to think about, we're talking here in the context of crop improvement and thinking about wheat, expected increase of 60% demand within the coming decades. There's estimates that if we don't do anything, and current practices, current varieties, with climate change, that gives us some major yield decrease. And put these numbers together, and you need a rate of gain at or above 2% per year. And if you look at most programs, most regions of the world, the current rate of gain is less than 1%. And so clearly there's a lot that needs to happen to shift from 1% to 2%, basically doubling our rate of gain per year. And so in the context of breeding, always think about this as accelerating, making this breeding cycle more efficient. And so how can we go from crossing to speed up to the evaluation and more quickly and more effectively do selection. And so if you put this in a quantitative genetics context, you get an equation of some form like this, which in the simplicity is you're just looking at genetic gain over time. So how much better is the average population this year than it was last year? And this is a function of the selection intensity. And so what portion of the population are you selecting? And of course, as John really pointed out, if you select too stringently, you quickly increase your inbreeding. And so this is limited. Your selection accuracy is obviously how close your phenotype or your genotype prediction is representative of the true genetics of that individual or that family. The genetic variance, obviously if you have no genetic variance, you're not going to make any gains from selection. So each of these parameters you want to try and increase and optimize. And then because this is gain over time, you have that as a function of years. So you could take 20 years and have perfect prediction accuracy and evaluate all your progeny in every environment. But because it takes so long, your gain per year, your gain over time will be really low. So we want to obviously minimize the number of years to go through one of those cycles of selection. And so just thinking in a historical context, thinking about the CIMMYT programs or many of the wheat breeding programs around the world, a lot of these things have been addressed. And so if you look at plant breeding in the last half half-century, there's been a really great increase in selection intensity. A lot of this fueled by new technology, split plot combines and things like this, generating really large F2 populations, big screening nurseries, many crosses, many populations. Also increasing the selection accuracy sort of in a statistical framework using replicated testing, multi-location testing, and this is to account for field and environment variation. And so with some statistical corrections, you can separate the genetics from the noise, and you can be making more accurate selections on your true underlying genotype. Same thing too, you talk about genetic variance, and so this is bringing in new genes not present in the program, or as was just pointed out, conserving the genetic variance that is currently there. And so this maintains that. And there's a lot of great examples just using germplasm resources. Also, this is a classic example thinking about reducing plant height. And until the semi-dwarfs were introduced, there wasn't much variation to actually do that and make selections. And then the cycle time. And so this is historically largely achieved by multiple generations or off-season nurseries per year. And so the example at CIMMYT with the wheat program is the shuttle program that Dr. Borlaug introduced effectively cut the cycle time in half. And when you cut this in half, you basically almost double your rate of gain per year. So when we think about genomic selection and precision phenotyping, these aren't really new concepts, right? These have been around. These gain from selections were recognized many, many years ago. And the only thing is we just have new tools. And so I talked about split plot combine. You can effectively double the number of plots measured. We talked about genomic tools that let us observe the genes directly rather than indirectly observing the genomics through the phenotype. And then stuff like sample tracking, and we'll talk about high throughput phenotyping. So just a real brief overview. Mark gave a good introduction on genomic selection, and I'll just briefly highlight how we're applying this in the CIMMYT programs in particular. But we have the training population and the selection candidates. These are genotyped and phenotyped, and then we just genotype on the selection candidates. And so one key to this is needing inexpensive, high-density genotypes, and I think this is largely achieved and put in place. And then the other caveat is having good, accurate phenotypes to actually train this prediction model. And so that remains to be a critical component. And so just an example, one of the favorite traits that we work on for GS prediction is actually wheat quality. And so you always have this balance. On the more difficult, the more expensive your phenotype becomes, the more advantageous, the more advantage there is for implementing a genomic prediction. And so you have these wheat quality traits. Some things like test weight, grain hardness are relatively easy to measure. They're fairly high throughput. When you have something like alleviograph and a low volume that actually require taking a sizable sample, milling it, making a dough and baking it, these are each individual data points, and obviously not a very high throughput phenotype, costing this whole set of phenotypes on a single sample would run in the order of $200 to $300. And so if we're able to take a $10 genotype profile and make a prediction on these, we can do quite well. And so this is just coming from a set of CIMMYT breeding lines over several years of nurseries. These are the same ones that Pancho talked about. And we can actually achieve pretty good prediction accuracies. And so these are actually a forward prediction, taking previous years and predicting into the next year. And if we are achieving prediction accuracies of 0.4 to 0.7, this really validates the ability and the utility of genomic predictions to actually pre-select what goes into these advanced nurseries and what goes into quality testing. Also, we have the ability here to greatly increase the size of the populations. And so in our current projects, we're genotyping 7,000 to 8,000 of the new breeding lines each year. We can generate a quality prediction for every one of those now, contrasting to the quality labs that are really limited to a few hundred samples per year. And so overall, we've seen this across a number of traits, and genomic selection giving reasonable to actually very good prediction accuracies. Here again, depending on the complexity, the quality of the phenotype, and obviously the populations that you're looking at. But this really enables selection on a single plant or a single seed. And so here again in these programs, especially for yield and quality and things like that, you really can't do any type of phenotyping until you have a sizable amount of seed. A big thing I won't talk about, but we're, especially in places like Kansas, where you have extremely variable environments from year to year, and you often lose entire seasons or entire nurseries, you can actually make selections in unobserved environments or years where you have no good phenotype data. There's also strategies you can put in place to maintain genetic diversity by selecting, sort of in a set theory, selecting sets of parents that will complement. And as I pointed out, this enables evaluation of much larger populations. But the biggest thing besides all of these is optimizing the top side. Genomic selection really enables decreasing the number of years to get to predictions. And so here again, the quality predictions are happening at much advanced stages. And so being able to do those predictions and selections early on in the process actually cuts many years off of the selection cycle. So overall, I think that what we've seen over the past few years is sort of on the technical challenge. You see a lot of examples of good prediction. And so technically, genomic selection, we can make good predictions, we can do the genotyping, and so really, and in my mind, it's sort of a logistical challenge remains of getting all of the pieces working together between the breeders and the genotyping lab and the quantitative genetics group and getting the predictions done and in the hands of the breeder at the right time. So just to summarize on that, and this just comes from an example of the paper we had in 2012, but we had prediction accuracy of 0.42 for yield under drought, 0.33 for 1,000 kernel weight, and 0.33 for heading date. And so these are like fair to reasonable. But the one thing I want to point out is and we've seen this in different sets of data across different breeding programs, is that what we presume to be very highly heritable traits actually end up having a lower prediction accuracy. And just to explain the reason that we really think this is, we see the same thing for plant height too. And the reason is we've trained all of our prediction models have been trained on the most elite sets of material. And at that point in the breeding program, heading date and plant height are largely fixed and very uniform. So you've basically exhausted all of your genetic variants by the time you've reached that advanced stage in the selection cycle. And so because we're genotyping all of these candidates, and most of these are actually in the field because they have to go through a seed increase anyway, we actually need to get good data for heading date, for plant height, for some of these agronomic traits to increase our prediction models. The trouble is, breeders don't like to take these kind of data because it's very easy to select one that's right. It's too tall or too short. It's very easy to select those out. It's much more difficult to get a quantitative measure of that phenotype that we need to build the prediction models. And so this will be a big challenge, and I think this is, as maybe you see as the rest of the talk, is when we can integrate this high throughput phenotyping to capture those traits, it's going to feed back and increase the prediction accuracy on all of these agronomic traits. So now to shift and talk about how this fits in looking at the current breeding schemes at CIMMYT. It's going through a selective bulk program from the F4. You're selecting single plants, and those, about 30,000 of those go to F4-5 small plots, okay? From there, about 7,000 are advanced to first-year yield trials. We're at the point where we're genotyping all of these 7,000 each year. And then 1,200 advanced, and then those are disseminated after a second year of testing to the international nurseries. And so what we're trying to think about now is how do we really optimize these first stages of selection? And implementing, like I said, we're implementing here selections for quality based on the GS predictions now. But how do we actually start to move this back up a generation? And so this comes from some modeling that Jessica Rutowski has done. And this is basically looking at different stages and places to do selection during those stages of testing. And so if we go from the bulk selections to stage 1 and stage 2, at each one of these you could select some portion of individuals and feed those back into the crossing block to intermate. The problem is that because you have limited levels of replication and then limited environments, your accuracy at this stage 1 is actually pretty low. So selecting parents for the next generation is sort of risky at low accuracy. And so what Jessica looked at was estimating genetic gain from improved selection accuracy in stage 1. So if we can combine genomic information, if we can combine correlated high throughput phenotyping at this stage, what's the effect if we can increase this selection accuracy? And so the approach she took was just to calculate accuracy for selecting a number of individuals from each one of those paths. You basically take a weighted average gain from each of the points of selection, and then an average time. So to point out that if you're selecting, here again we're trying to reduce the cycle time. So if you're selecting individuals at that first year when they're in the field, you obviously have 2 years shorter than if you wait until year 3. Even though the accuracy is higher in year 3, you have 2 additional years to meet that point. And so the simulation approach was just to simulate all combinations of different levels of selection at each one of those stages, estimated genetic gain for each one of those combinations, and then find the maximum to optimize those. So you vary the population size, you vary the portion of parents selected at each stage. And so just 2 slides here showing some of her results. The first was stage 1 accuracy. Obviously, if you increase that accuracy, your genetic gain is going to increase because you're selecting more and more individuals from that first stage. And actually, you see here, current accuracies are probably on this low end here. And if we're able to push that accuracy up to 0.6, you're having a very, very large increase in your rate of genetic gain. And this is on a per year basis. And so what's that look like then on the number of parents you're selecting? So in this current scope where we're operating, you're basically selecting some number of parents in stage 2 and some number of parents in stage 3, and this is pretty consistent with what I understand is going on with Robbie's selections. If we can actually use some methodology to increase those selection accuracy at the stage 1, increase that selection accuracy to 0.4 to 0.6, you actually select a substantial number of individuals from that first year of testing. So how do we do that? Obviously, what's currently is you do phenotypic selection. You go out there and visually look at them. You look at the seeds. As I noted, what we're implementing on some traits is genomic prediction. And what I'll talk about now is actually incorporating high-throughput phenotyping plus a pedigree-based approach, because remember, at that stage of 30,000, we're not yet at the point where we're genotyping all of those. But you can, as was pointed out, you can use a pedigree-based genetic relationship to get a similar type of prediction. And so some of the things we can measure in fairly high throughput are NDVI. This is normalized difference vegetation index, basically a measure of greenness or green leaf of the plant. We can measure canopy temperature, and then we've got a lot of work to take these other physiological traits that have been well defined and to put those into a high throughput phenotyping context. And then kind of in my mind, we'll come back to this, but thinking about, okay, let's just throw everything together and try and push this as far as we can. And so just to give an idea, this is the correlation between some of these physiological traits and grain yield right here in this column or this column going down. And you see, so NDVI during grain fill and during vegetative has a fairly strong positive correlation. Canopy temperature has a fairly strong negative correlation to grain yield at each of those stages. And so both of these, obviously selecting ones with a higher NDVI and ones with a lower canopy temperature, can be used as correlated traits to help assist in generating more accurate predictions and selections. And so we're demonstrating this now, but a really nice, there's been several nice demonstrations of this and success in animal breeding. This is just one example of a paper, and really what came out of this was that the inclusion of correlated traits, so like I was saying, NDVI and canopy temperature, including these correlated traits greatly increased the prediction accuracy, both in using a genomic and a pedigree-based prediction model. And also as the number of traits increased, so this is what I was saying, the number of correlated predictor traits, as that increased, the advantage of using genomic rather than pedigree relationships decreased. And so with a number of correlated secondary traits, you can actually help push these pedigree-based models up close to the prediction accuracy achievable with genomic selection, with the genomic pedigree. And so just to talk about how we're starting to apply this now, this is in the context of Feed the Future Innovation Lab that's with our group, Cornell and CIMMYT, working across the BISA locations and with the Pakistan National Program is supported from USAID. But here's just a heat map of wheat production here in South Asia. And these are the testing locations that we have for this project at Jabalpur, Pusa, Ljubljana, and Fauslabad. And so this is also representing, one of the main targets in this is thinking about maintaining yield and production under heat stress. And so you also have this gradient of heat stress as you go from really optimal wheat growing conditions in the Punjab, and you have increasing heat stress as you head south and east from there. And so just one of the things we've done in a very simple way to sort of start this is to take and using, this is just a handheld green seeker measuring NDVI on these plots. This is early vegetative, and we did this all the way through heading here, is to build models to look at the rate of senescence using basically these correlated predictor traits. And so across these three locations, you can really see this is the average of the overall population, but you can see very different response curves based on the timing and the duration. Largely, this is because these are under optimal irrigation. These are largely due to varying heat stress across those locations. And so we can build these parameterized models now, and we're in the stage, and we're in the process of taking these traits, taking these models, and then incorporating them into the genomic predictions. And so we've been able to do the same thing using those measurements. I didn't say, but we have NDVI and canopy temperature measurements every 10 days across that growing season. So even with that level of data, you can build pretty nice regression curves across each one of those entries in the trials. And you can really see the dynamic differences both across the locations and between the different genotypes and just how they're responding during grain fill to those heat stress. And so really the key now is to say, okay, we've got the contextual, we've got the context to use this high throughput phenotyping to integrate it into both just a pedigree and a genomic prediction model. And so now we need to make it high throughput so we can easily assay those 30,000 plots and also the tens of thousands that are in the advanced trials at CIMMYT. And so we've been doing a lot of work, just to introduce briefly and conclude here on some of the work we've been focused on high throughput phenotyping. For the ground-based stuff, we've been taking what we call a geo-referenced proximal sensing approach. And so to integrate a high precision GPS that will give you centimeter level accuracy with these different types of sensors. And so obviously the first ones we put on there were something that will measure NDVI, something infrared thermometers that will measure canopy temperature. And so with the integration of the GPS, you can precisely place each one of those sensor measurements within a field plot as you traverse the field. And so this is just a visual of what some of that data looks like. Here's one of the fields that we ran. You can see the low NDVI values. This is latitude and longitude, and you can't see the individual data points, but there's tens of thousands of individual measurements here. And you can see here's basically bare soil at the end of the field, and you can see variability across the field, but if you zoom in on this, you basically have continuous sensor measurements. These low values are the bare soil between the plots. You can define plot boundaries and trim those, and then based on our row column field design, you can assign all of those data points back to individual entries in that experiment. So we've been using this context and approach to do some good high throughput phenotyping. And so what we're doing now in the context of these projects with CIMMYT and BISA in the innovation lab is basically transporting that here to India. And so this is what we call PhenoCart. The one we built in Kansas is actually a really large motorized vehicle. This is our portable version. This is Jared Crane, and this is at the CIMMYT research station in Obregon. But basically built this cart that incorporates all of the same principles. We have differential correction GPS, we have the sensors mounted here, and all of this data is just logging to a PC. And so this is measuring some of the fields at BISA Ludhiana, and this is some of the data that's just come back in the last few weeks. And so now we're taking these physiological measurements, making them high throughput, and then we'll be able to integrate these into the prediction models. And so the other approach we're taking on this is an aerial attack using these UAVs, unmanned aerial vehicle. The platform we really settled on is this 3DR Iris Plus, primarily because it's inexpensive, less than $1,000. It's very user-friendly and so easily deployable. We're getting a whole bunch of these units, and just in this example, we're using an NDVI camera that was converted from a Canon S100. And so with this whole setup, that system is probably I think it's less than $2,500 for this total setup. Just some examples of the images coming off of these. This is, again, some of the work we've initiated at the BISA sites. And this comes from just over the past few weeks. These are false color NDVI images from Ludhiana, and we're now able to extract on a plot level each one of those pixel values and to get a really accurate assessment of NDVI across those plots. This is just a bigger scale. You take hundreds of those images, you stitch them together, and you can get your entire field nursery of, this is about 1,500 plots at the current nursery in Jabalpur. And so, as Prasanna nicely pointed out, now our main work has shifted into data analysis. And so, just as it's become very easy to generate genomic data, it's becoming easier and easier to generate phenotypic data. And so here again, the major effort and the major constraint shifts into bioinformatics. And so, to conclude here, just to bring this back around and think about increasing the rate of genetic gain across these breeding programs, I was kind of pointed out how both high throughput phenotyping and genomic predictions integrated together also can increase the selection intensity by enabling these much larger populations. And so now we're talking about implementing selection on thousands of individuals for something like grain quality, and tens of thousands of individuals for something like yield. We can increase those selection accuracies both in the genomic and through integrating these correlated high throughput phenotyping traits. And with this, and here again, so the major challenge will be to how can we maintain and increase this genetic variance. And both through the genomic and through developing basically phenotypic prediction models, right? Using these correlated traits, you can show how you can decrease the number of years needed to advance through a cycle of selection. So with that, I'll just thank, I've got a really great team and great collaborators on all of this. Shuangye Wu is a technician in my group who does all of the GBS and generates the genomic data for all of these large projects. Jared Crane is a student in the group who's built this phenocorn and really instrumental in deploying that. Daljit is a student in my group who's running the UAVs right now. And then obviously the great group at CIMMYT, Ravi and Suzanne running the breeding and the molecular breeding programs. And Ravi Valaru and Uttam have been instrumental in managing the field sites here in India. And then Jessica has done a lot of really great modeling work. And so these projects are largely supported from USAID. Our high throughput phenotyping work is supported from NSF, and also these projects are supported by the Kansas Wheat and the Gates Foundation. So I'll stop there and take any questions you have.
Thanks very much. We will have just one quick question from Dr. Siddiq.
Thank you. Would you tell me, when you have such a large number of genotypes in the field, how you're going to adjust for the differences in phenology, for example, the canopy temperature, NDVI, and so on.
Yeah, so that's, so that's what we started to look at on parameterizing these models if we need to adjust them. So these were all adjusted to actual heading date and then thermal time past heading date, just on the example in the senescence. But then you get into the complexity of depending on when they were flowering and optimizing that. And so I think I don't really have a good answer at this point. I think we'll have to sort of empirically investigate and try different models, some that are maybe adjusted on phenology and thermal time, and some that are maybe just based on actual calendar dates, because you obviously need to optimize both development and phenology.
Okay. Thank you very much. And if you have other questions, catch him during the lunch. And we will close this session. Maybe.
Thanks very much, Dr. Siddiq and Dr. Kaur, for chairing this session.